go. Hey, welcome back. I changed my hat. Okay, we're back to here. Phosphate backbone. These are your bases. In the middle, A goes with T, you have a double bond. C goes with G, you have a triple bond. Here's your 5' down here. This one's upside down. This is how they connect. Okay, let's move on. The two strands of DNA are said to be anti-parallel. Here, again, you have your two strands connected by thymine and adenine, and your 3 bonds connected to cytosine and guanine. And it's your purine always with your pyrimidine. Each pair of bases, oh, I think I skipped one. Oh, each pair of bases are connected by hydrogen. This should be hydrogen bonding. So these are hydrogen bonds. Remember I said if you need to guess hydrogen bonds. Seriously. Adenine and thymine form 2 hydrogen bonds. Cysteine and guanine form 3 hydrogen bonds. Just what we said over there. Okay. The pairing of the bases are complementary. So whenever you get a thymine occurs, an adenine falls, we're opposite of it. Where there's a cytosine, there's always a guanine. A goes with T, C goes with G. Do we have that? Oh, that's what complementary means. A goes with T, C goes with G. They complement each other. You complete me, guanine. Do you see how that works? And it causes this twisting. It really does cause this twisted shape right here because you get A's and T's and C's and G's. Okay. Duplication, transfer, and expression of genetic information occur as a result of 3 fundamental processes: replication, transcription, and translation. Replication is just making another copy of your DNA. Transcription is going from DNA to RNA, and translation is from RNA to protein. Okay. Replication is the process by which you replicate an identical copy of DNA. Transcription. This is the same language, you're just going from DNA to RNA. Translation is the process where you get these codons, do you remember? And the codons are connected to a protein, and then you make a string of proteins. That is translation. Ooh, my goodness, that is a big, I don't know what that is doing up there, but it's great. Okay. DNA replication begins at the nucleus with a partial unwinding of the helix, and this is called the helicase. So now we're replicating our DNA. This little helicase comes in here and it separates something. It pushes it apart, it shoves, these are together, all wound up, and the helicase comes in and opens them up. It just pushes the opening. That's what the helicase does. The unwinding occurs spontaneously in many specific origins of replication. So there's specific spots where the helicase is going to start unwinding, things are going to do the replicating. The origin of replication is the beginning, where it's going to begin. DNA strands separate, exposing what looks like a bubble. If you watch it in real time, then you'll see there'll be just where it separates, there'll be a bubble looking thing. It looks just like this. It starts to bubble, and this is replicating in here. There looks like a little bubble in there. DNA replication begins in the nucleus. At either end are branch points known as replication forks. So the place where it's pulling it is known as a replication fork. The template strand can only be read 3' to 5', and the new DNA strand can only grow in the 5' to 3' direction. So this is called the new strand, one strand is the template and the other is the new. So this isn't the best graphic. These are both green. You can see that this is the old strand, this dark green one, and the white strand is the new strand. So there's one old and one new in each new strand of DNA, in each new double helix. Only the leading strand can grow continuously. Remember I said 5' to 3' is super important. So this one's properly connected. This is the head, 5'. This is the tail. So it grows this way, and it can continually, it bubbles out, and it starts replicating right here, and it's all good. This guy can only grow 3' to 5'. So it has to go in little short jerks all the way backwards. So there's a leading strand, which is the main strand, and it's able to grow continuously. The other strand is the lagging strand. It goes in the opposite direction, and it is replicated in short fragments called Okazaki fragments. The Okazakis, it's a husband and wife team, they discovered this, and we named it after them. Well, I didn't. But they named it after the Okazakis, because they're the one that discovered that this happens. This one's happening continuously. This one's happening in short little bursts going the opposite direction. The lagging strand is replicated in short segments called Okazaki fragments, which we already said. And then it's stuck together. See, here's the short fragments. And then DNA ligase, kind of like gum, ligate, it puts it together, comes and glues it together with strands of DNA. So this will be a smooth strand in the end. Ligase is the glue, the gum. And this is the picture in your book, and you should be able to explain this. If it shows up on a test, if it shows up on Sapling Learning, you should be able to explain it. Yeah, I just explained all of it too. I think we want that. Okay. RNA has 3 main functions. Messenger RNA. This is our friend, our favorite, this little messenger RNA, this little mRNA comes here. You have your DNA, it goes in and makes a little strand, and then you have your messenger RNA, and it travels from the nucleus out into the cytoplasm, finds a ribosome. So mRNA, messenger RNA. You have your tRNA. This is the fun tRNA that has the codon on it. Remember we did the puzzle in class? It had a little codon there. mRNA versus tRNA. mRNA is going to go in here and take a copy. tRNA is going to have a amino acid on it, and it's going to actually make the protein. Ribosomal RNA. Our RNA is basically this big giant blob. This sandwich blob here that actually squishes the tRNA and the mRNA together and makes this code come together. This is our RNA. Say it 5 times fast. Our RNA, our RNA. In transcription, it's very much like replication, except it's DNA to RNA, and only one side gets translated at a time. It's basically the same thing. A small section of the DNA unwinds, the bases are exposed, and complementary nucleotides attach. The DNA strand that is transcribed in the template strand, its complement in the original helix is the informational strand. We call it an informational strand if it's going to be the DNA to RNA. So is the template strand. Its complemental original helix is the informational strand. The mRNA is an exact duplicate of the DNA template strand, except that U replaces T. So it's exactly the same, except there's a U instead of a T. RNA polymerase moves down the DNA segment, adding complementary nucleotides on the growing RNA strand. Again, very similar to DNA replication. Transcription ends when an RNA polymerase reaches a termination sequence. You will see this, a stop codon, when we do codes. You'll see some of them say stop. That's a stop codon. It says, okay, stop doing stuff. So here you go. DNA. Here's the DNA. Here's the triplet code. It makes the RNA right here. Do you see? It makes an RNA. Then this RNA goes off with a codon and goes to make proteins in the cytoplasm to find the ribosome. Does that make sense? DNA makes a strand of RNA. RNA goes off. Okay. Only about 10% of base pairs in DNA code for genes. 10%. These fancy little introns and extrons. The code for a gene is contained in one or more small segments of DNA called exons. This is important. Exons exit the nucleus. Exons are the little pieces. Here's an exon. Here's an exon. Here's an exon. We're going to splice those introns out. We're going to make one with just the exons because they exit. The introns don't do anything. Okay, my 10 minutes are up. I'll see you in a minute.